processes here, and if you really spend some time on this and understand this, what we're going to talk about here, you will understand how hundreds of drugs work, okay? And all you have to know is say, what does a drug do? Well, it's a calcium channel blocker, it's a sodium channel blocker, it's this or that, okay? And you'll know exactly how they work, okay? So, remember, I just need that back after you're done with it. So, and then I'll show you a little video that sort of ties it together a little bit too, so if you're visual, you can see how things are working and so on. But we're going to talk about muscle contraction, that apparatus, the proteins that do that, and then we're going to start over here with the way we control that, with nerve conduction, and how the nerves stimulate the muscle to get the action potential to go, and that stimulates the contraction. Okay, so it'll be a big, long, loaded process here, okay? But one of the simplest things to get over it, you remember that video I showed you with the little walker? Walking down the microtubule, okay? That takes ATP to cock that on, right? It's an electrostatic repulsion. What our body has done, and mammal muscle cells have done, is take a very similar protein. Now, that was just one little protein, dragging a vesicle, walking on a microtubule. But it's knocked off one of the feet, okay? So now I've only got sort of one walker, all right? But how do I make a muscle out of a walker? Okay? Some engineer has to think about how to take a walker and a fiber and make a muscle out of it. How would you get a cell to contract? What would you have to do? What if you just had that microtubule and one walker? How would you do it? Well, it's going to have to drag something with it, though, right? Well, ATP is floating around.
Is that where the potassium comes in?
Well, potassium's not going to make the cell contract.
Okay.
We have to take this mechanical structure, and somehow we have to hook this to one wall, hook this to the other wall, and then the cell will contract as it walks, right? It'll get smaller. Okay? So that's the theory behind what's going on with our muscles as well. There's a lot of people actually think that simple arrangement, just the microtubules, either actin or microtubules, and those, they're called dyneins and kinins, they have all sorts of names, but they're very similar. These motor proteins, these walking proteins, they think that that's how simple cells, like when you have, say, a blastocyst, an embryo, okay? It's just differentiated stem cells, okay? They're not muscles, okay? How does that thing in the womb all of a sudden invaginate and form a cavity, which then goes all the way through and forms the gut, the mouth and the anus? That's how it forms. It just folds in on itself like that. How could cells do that? They don't have, the cilia really, they're not moving or anything. But they think that that's how it happens. It's just simple things like actin and motor proteins that are causing the top of the cell to cave in, and then the next one, the top caves in, and that creates a curve. Okay? Just like an arch of stones. They're all curved more so at the bottom, or at the top than at the bottom. So when you pull the sides in a little bit, that causes the whole thing to invaginate. Okay? So that's a real, ancient form of motoring, moving. Okay? But now, evolution has given us something a little better. Say I have a wall here, and then I have another wall here.
It's like you're tearing down the walls.
This would be what we were looking at as microtubules, but in this case, in our muscles, this is actin. It's a monomer. Remember that polymerizes and forms that actin fiber that has a lot of the bumps on it, okay, almost like a ratchet. Okay, so that's what these things are here. They're called the thin fibers of this structure. And I'm not going to do too much nomenclature necessarily. I'll spill out a little bit, but you don't really have to take notes on this right now if you just want to watch and listen, because I'll do it again. You have it on your slides, so I prefer you to get the concept than worry about the names right now, because you're not going to miss anything. We're going to go over it again, okay? this whole little muscle-like structure here is called a sarcomere. And anytime you hear sarco, like sarcoplasm or whatever, you're talking about muscles. Okay? They're called thin fibers. They stick in, they're attached to a structure. Okay? If this was a real simple muscle cell, it could be the walls of the cell itself. But our cells have thousands of these things all lined up. So there's walls and walls and walls and walls and walls. They're all hooked together. So one of these units is not a muscle cell. It's thousands of these things stretched out in fibers, and they all contract at once. Okay? But the walker, I could make a walker like this. Okay? Put the little feet on there. The original one we talked about only had one foot, right? Well, what our body has done is taken hundreds of those things with the walkers and wrapped them around each other in a spiral-like thing so all the walkers can work together. So you've got all these heads all down this fiber. Okay? So I'm just going to draw it like this. It's called the thick fiber as opposed to the thin fiber here. Okay? And it has names. I'll talk about that in a little bit. But instead of one direction, okay, the body does it in stereo. It makes another one on the other side over here. Okay, so it has walking feet. All right? I'll just draw another one just so you see this pattern that's developing here. I'm just going to do this. Okay? Those are all the walking feet. Lots of them. Okay? But you can see you're starting to develop the pattern in these bands like this. Now this thing, when it fires, all it needs is ATP to actually work. Okay? So if we dump ATP on it as a trigger, they'll start walking. You can see what's going to happen. Okay? All these ends, they're going to pull those strands in, and the walls are just going to move in, right? Okay? So, and that's what's going to happen. This thing is going to collapse. But we can't really use ATP as the trigger because we can't sequester ATP and then dump it on the muscle and get it to go. Okay? Because ATP has to be out everywhere for all the other enzymes that are working. Okay? ATP is just ubiquitous, floating around. So this thing, if it wasn't controlled, the ATP available would just cause these things to walk to the end and then just stop. Okay? It would be contracted, okay? Which is basically what happens in rigor mortis. When the whole control system breaks down, the triggers are let loose, all the muscle cells contract, and you get that rigor until the muscle starts deteriorating and then it loosens up again. Okay? So there's our system. Okay? Based on, they're all proteins. Okay? No sugars in there. We don't need glucose to make it work. Anything. All we need is ATP. So I'm just going to put ATP out here, floating around. Okay? And when it bonds to each one of the heads, okay, every one of those heads, every time a head just goes cocks and makes a move, and ATP is used up. As you can see in your muscle cells, they're using millions of molecules of ATP per second. I mean, probably billions. Okay, so the mitochondria are really kicking the ATP out. And there's a fair amount of mitochondria all along the muscle cells. Okay, so inside the cell, so they're kicking that out. So we can't use ATP as the trigger. What can we do? Well, it turns out that it's kind of an interesting little process here. The sites where the walkers walk on, that ratchet, the actin here, okay? If we block those sites, the walkers can't do anything, okay? So what your body has done is taken another protein and wrapped it in spiral fashion around the actin and blocking all the sites that bond, hydrogen bond, that's how they bond to the actin. It won't work. It's blocked, okay? So I can just sort of put a little spiral thing here like this to show that. Okay. So it is getting a little more complicated, okay? Okay. Now, how do I take the spiral off? How do I make it go away or move or something like that, okay? Well, there's another little protein that hooks to the, call them the peaks if you want to, okay? Separate little protein. I know I'm writing this pretty small here, but I guess you're getting the idea. That little thing will bond calcium. And when it does, you get electrostatic. Calcium's a double positive ion. You get electrostatic repulsion, and it causes the whole thing to untwist, and then the sites can walk. Okay? So calcium is our trigger. Okay? So that's CH or Ca double positive, not Ca. Okay? So that's our trigger. That's when we dump calcium all over this whole unit. That's why it moves. Okay? ATP is just there anyway. Okay? Yeah.
So they always make a big deal about calcium with for your bones,
Yeah.
but it's also very, very important for our muscles.
Oh, it's vital for your muscles and your nerves. Your nerves won't fire without calcium, and I'll show you why in a second. So, yeah, calcium. And if your body gets low on calcium, it will deplete the calcium in your bones. Okay? You start releasing hormones that cause your bones to dissolve. That's why you get thin bones if you're not getting calcium in, because your body is using it. And you're always losing a little bit in your stool and in your urine, so you need calcium to replace all that, to keep the strong bones.
Is that why they say that calcium for the bones, because your bones what gives it up?
Yeah,
for
it's
everything
basically
else?
calcium storage, and also phosphate storage. Calcium phosphate is the cement that holds your bones together. So if you need phosphate for ATP making, or if you need calcium for any other reason, the hormones will just dissolve it. The osteoclast are cells that just dissolve the bone, and it just floats away.
Wow.
So, all right, now we need to figure out how to release this on command. Okay. So it turns out that we have a sack of it on here, magically. Okay. Lots of calcium in it. And it has a trap door on it. I'm going to draw it over here. You'll see why in a second. I'm going to draw this as sort of a pore with a trap door on it. I've got a lot of calcium here because I've got active calcium pumps out here. This calcium is being pumped in with ATP. That's an active transport. Okay? So you're pumping it against its concentration gradient. It's getting real concentrated in here. And all I need now is a trigger to dump it out. Okay? And then when I stop that, these will pump it back in. And this happens in milliseconds, thousandths of a second. Boom, contract, and then sucks all the calcium back. So it really works fast. Okay? But so now we need a trigger for this door. And this is just going to be a fast diffusion. It doesn't need to pump it out. Okay? Because this is concentrated. It's just going to flow right out when you open the door. Okay? So we need a way to open the door now. Okay? And on the outside of the membrane, I'm going to pretend like this is the cell membrane. There is an extension of the cell membrane that comes down. Okay? This is called the T-tubule. Okay? This is the sarcolemma, the membrane of the muscle cell. Okay? And it's that lipid bilayer, phospholipid bilayer, just like all membranes. It comes down and forms a tube. Okay? It's not quite as straight and pretty as this, but it's coming down through. And I need now to get something down in here or do something to trigger this to open so that the calcium can flow out when it's triggered. It doesn't flow into the T-tubule. It actually flows out in a little space right here. Okay, there you go. Okay. And so what's going to do that? Well, that's where the electrical charges and ions come in. That's our messenger. It's positive sodium ions.
Is sodium?
If I can get sodium ions next to this door, okay, positive ions, it will open.
It will open.
Okay? And so now I need to figure out how to get sodium ions there on command. Okay? And that sort of makes us want to go back and start at another story here. How do we get membrane potentials with positive ions coming down into the membrane? Okay? So let's leave this picture here for a second. We're going to start over here with our old friend. What do I draw? Well, for obvious reasons, I'm going to do something like this, and then I'm going to come over here. And this is all going to be connected. Okay? This is our muscle membrane. Okay? And over here, our old friend, the sodium-potassium pump. Okay? That's going to put a lot of sodium on the outside, much higher concentration on the outside, and we're going to have potassium on the inside, okay, in much higher concentrations. So what? That's not really doing anything because the charges are balanced. There's no electrical charge on this thing right now because all of these sodium ions are balanced by chlorides and phosphates, and same down here. Okay, so there's no charge difference. There's a concentration difference between the two ions, but there's no electrical difference. Okay, so they're in balance. Now we have this weird thing called the potassium leak. Okay, potassium is slowly leaking through this membrane or protein. Okay, and what that does, if you only allow something, it's a selective leak. You only allow potassium to leave. As soon as one potassium ion leaves, there is a charge balance now because whatever was balancing its charge, like a chloride or a phosphate down below, is now causing a negative charge below the membrane, and now we have a positive charge above the membrane. Okay? So when potassium leaks, and it doesn't take much to leak, I start to get a positive charge just on the very surface of the membrane. It doesn't go very far, just on the very surface. And I start to build up negative charge underneath. Okay? I'm going to just erase that one for the time being. So now we have an electrical potential. We essentially have a battery. Okay? Anytime you put electrical charges separated with a potential to get together, that's a battery. That's an electrical potential. It may not seem like much when you just have a few ions under the membrane or above, but the distance is what's important. One positive charge like this across the distance like that, if that was the size of a car battery, it would be 1,000 volts instead of 12. Okay, so these charges really do make things move. Okay, one little positive charge makes proteins, move. They're either repelled or attracted depending on how they're made or how they're built. So this is the important concept here of setting up an electrical potential based on a sodium and potassium concentration gradient problem. Okay?
That's a question.
Yeah.
Last test, whatever you want to call it, you had told us that the potassium channels, basically without those potassium channels, that your muscles didn't contract, your nerves didn't conduct. That's why?
It's only part of the reason.
Only part of the reason, okay.
Yeah, we're building up to the switches.
Okay.
Right now we have a battery with no switch.
Okay.
Okay? How could this change?
Okay.
This would be an equilibrium. Okay? And what happens is, if we actually took a voltmeter, okay, and we put a meter on the outside and one on the inside, just above and below the membrane, and we measured the voltage difference, it'll look like this. Okay? It's about minus 70 millivolts. Okay? So if that's minus 70, let's say that's 0, let's say that's positive 70 here. Okay? Millivolts. And this would be time, because this is going to change. Okay? So this is where we're starting. You can't really see it minus 70, can you? Don't worry about memorizing the numbers. Okay, I want you to the concept. So below the membrane, we're at a negative voltage, okay, based on our little voltmeter. Okay? So that's where we start. That just happens to be the voltage that potassium leaks out. And you can see it's going to leak until there's enough negative charge down below here that it can't leak away from it. Okay? Once the charge gets negative enough, that it's pulling on the potassium, and the last one just can't get out. Okay? So it reaches an equilibrium at minus 70 millivolts. Okay? So it's not going to leak forever. Okay? Now the great thing is, now we've got a negative charge under the membrane, the whole membrane. Okay? It is now negative, 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 negative here for the exact same reason. Okay? Because those things aren't just over here. There are thousands, probably millions of them all over the cell. Okay? So the whole cell is polarized. Okay? And that's how you, and that's what we got to change. Okay? So this little wavy part right here, this just happens to be the part of the muscle cell that is going to be attached to a nerve. Okay? And we'll get to those triggers after we talk about this part. I'm going to have another special little pore here. I'll make it a little bigger here just a second of that. This is what's called a chemically gated sodium channel. Okay? I'll write the word chem gated sodium. Okay? And what's going to happen here, you know, we've got a lot of sodium outside here, okay? As soon as I have a chemical, a special chemical that bonds to that, some sort of transmitter, okay, this thing will open and dump sodium inside the cell, okay? So I'm just going to put this thing, there's my little trigger, okay? When that gets triggered, sodium will come in, okay? And I'm just going to put positive charge around here like this, so you can see that everything else being negative, right? So in that area, I now have sodium just loves to come in, because sodium is not only going down the concentration gradient, it's being sucked in by negative charge. Okay? That's attracting it. Okay? So it just dumps in. And you'll get a little wave of positive charge here for sodium. Okay? And the great thing that's come that makes our nerves work and makes the membrane on the muscle work is that the next one over here is a sodium channel. It's not triggered by chemicals, it's triggered by the voltage. Okay? So it's a little different kind of channel, but it's still sodium. And that, when this positive charge hits it, it opens and dumps in more sodium. So you can see this wave of positive charge is going to move down the membrane, because these channels are all along the membrane. Okay? So one, the chemical one is the trigger, okay? But it triggers the voltage one, which then triggers the next voltage, the next voltage, the next voltage. And if we watch what's happening on the voltmeter, we'll see something like this. If I don't have enough chemical trigger here, I'll be moving along, and I might get a little positive as some sodium comes in, but if I don't get enough to trigger this one, it's going to go back down and nothing happens. Okay? If I get enough sodium in, it reaches what's called a threshold, which is about minus 50. Okay? Once it reaches threshold, boom, all these start to open, and you get a big rush of sodium. Okay? So it goes, the voltage goes positive on our meter. Okay? Now that can't last forever. Okay? What happens is when these voltage sensitive channels, when they get up to about plus 50, okay, they shut off. Okay? And so I'm just going to put sodium off. Okay? And right here, I'm going to put sodium on. Okay? And it's this voltage gated sodium channel that we're talking about. And I'm going to put voltage gated. So there are two different kinds of channels. They're very evolutionary. They're very similar. They have very similar patterns in their helices and things like this. So they're built pretty similar, but they're different in the sense of how they're triggered. So this can't last forever. So when the voltage hits about 50, they turn off. Okay, and what happens is a potassium channel turns on. Okay, and as the potassium then rushes out, okay, right there, we're going to go back to being negative. Okay, because the potassium is going to take positive charge with it, leaving behind negative charge. Okay, so we get boom, the sodium's rushing in, it stops, and then boom, the potassium rushes out. And as soon as that potassium turns on, the voltage will go back negative. Okay? And this is how you get the curve of what's called the action potential. Okay? And as soon as you get back down to somewhere in this range, these voltage sensitive potassium channels turn off too, and then everything's essentially back to normal. The sodium potassium pump over here will pump out the excess of either way, you know, and then everything gets back to normal. And this all happens in milliseconds. It's really fast. Okay? Think about how fast it has to be to get my toes to work just by thinking about it. You know, just lifting my foot like that. That's a millisecond. That's not even a second. Okay, but I can stop it in that time too. You know, my foot just doesn't keep going. You know, I can jerk it and stop it. Okay, that's less than a second. That's milliseconds, and thousands of muscles involved. Okay, so you can see how complicated and intricate it is.
So what would a spasm be? Something that's uncontrollable? What would be happening within the muscle?
Lots of reasons, because all this could screw up. You know, anything that causes more signals or less signals, you're going to get a flaccid paralysis or spasticity or seizures. I mean, that's why this is so important to understand. because all the drugs you can think of work on these channels. So now you can imagine that wave of sodium coming in, giving that positive charge, turning this positive, this side to positive 2, okay? That's going to trigger the calcium to be released. Boom, we get a muscle contraction, okay? And as long as the nerve here is giving us the chemical signal, okay, as long as it's stimulating this part right here, which is called the motor end plate, okay, of the muscle, as long as that keeps happening, you'll keep getting waves of positive charge, and you'll keep getting this cycling calcium thing going, so the muscle will contract and contract and contract and contract, okay? And I can stop it at any time, just, right, boom, right there, I can stop it, or I can keep on going, and that nerve is firing, telling the muscle, keep going, keep going, keep going, you know? So that's how it works, okay?
So when we stop our muscle from going any further, we're actually stopping the channels?
You got to stop this.
Stopping the nerve.
You
And
gotta
then the
stop the
nerve will
nerve.
send the signals to stop the pumping.
Right. If you block this, you get no signal.
You get
okay?
nothing after that.
And that's just a normal muscle tone. Okay. Your muscles are actually always being stimulated by your brain to some point. You always have some muscle tone. Okay. They're not completely paralyzed. Okay. They don't even feel paralyzed. You know. There's some tone there. Okay. So they're always being stimulated a little bit. and inhibited a bit too. Okay. You'll notice that when people have strokes and they knock out the motor portion of their brain that controls your arm motion, let's say, when you knock out that portion, you have upper motor neuron problems, and there's less inhibition, so that you're now paralyzed, but you're hyperreflexive, because the nerve is not inhibited like it is normally. Okay.
What chemical triggers that? Does a nerve send a chemical to that gate to trigger it?
Yeah, we're going to go over that right now, because that's our final key to causing this whole process to work. So I'm going to draw the end of a nerve coming in. Okay. This is an axon. This is called the button end of the nerve. This whole thing is called the synapse. Okay. The space in here is the synapse. Now we have a neuromuscular junction. Okay. The muscle side is called the motor end plate. Okay. But now we have a neuromuscular junction. And so something has to get out of that nerve to trigger that thing to open. And it's stored in vesicles. Okay. And there are little proteins that bond these things to the wall. Okay. They have special names, and there's half a dozen of them that do this. But the vesicles just don't bump into the wall and release their stuff. They're attached to that membrane, waiting for something to happen. Okay. It's basically, these things act like a purse string if you want to. It waits for a signal, and then it just tears it open, okay, and dumps it out. So that chemical is going to be coming through there, okay, when the vesicle is bonded to the wall, and it gets a chemical signal to tear it open. That chemical signal is a calcium channel, okay, and that calcium channel is voltage gated, just like this one and this one is. Okay, so when the nerve, which is negative, sends its positive signal down, the sodium is going down, just like in this one. It's a sodium wave of ions. When that gets positive, right here, it turns on this calcium channel. The calcium comes in, bonds to this synaptobrevin protein here, and that pulls the vesicle open and dumps out the chemical. So that is an important problem with nerves. Okay, a lot of calcium channel blockers affect your nervous system because of this. Okay, what is the chemical? Okay, acetylcholine is the name. It's common enough you might have even heard of it before. Okay, I'm going to put ACH for acetylcholine, and I'll write it out for you. I sometimes write all capitals, sometimes just one of my quirks. Acetyl, you remember the acetate molecule? Vinegar. There's vinegar, basically. We put an H there, you've got vinegar, okay? But this is now an ester. It's bonded to 2 carbons and then an amine with some methyl groups on there. This is the choline part. This is the acetate part. That's why they call it acetylcholine, okay? This is the only neurotransmitter that your body uses for your skeletal muscles, okay? This is every axon, motor axon that is attached to every single one of your skeletal muscles, okay, uses acetylcholine as the transmitter here, okay? So, but that gives us another engineering problem. And if you can think, well, we fire the vesicle, they dump the acetylcholine, they're stimulating the chemically gated sodium channels, okay? How do we get rid of the acetylcholine to stop that? Okay, remember, we got to do it within milliseconds. Okay? There are enzymes in here, and I know this is getting busy here. I'm going to color it in. Okay? This is called, for short, you can just call it cholinesterase, but it's acetyl choline, I'll start to write here, choline, and then it's esterase. You're breaking up the ester, okay? Ase means it's an enzyme, right? Acetylcholine, esterase. Now we've got some cursive writing in here too.
Keep it interesting.
Yeah. This is an extremely important enzyme because just guess what happens without it?
It keeps going, keeps them from.
Seizures. That's right. Continuous muscle contractions causes seizures. And that's what kills you with nerve gases. They are acetylcholinesterase blockers. Okay? Things you might have heard of like sarin gas and all these, military nerve weapons, and almost all of them are, if they're not, poisonous things that destroy your lungs, like phosphine and phosgene and chlorine gases, things like that. Most of the chemical agents are nerve agents that go after acetylcholinesterase, okay, because it works great. Okay, and you can see what's going to happen. There's something called a sludge syndrome that you get with low dose exposure to things like insecticides. A lot of insecticides are acetylcholinesterase inhibitors. That's why kids who drink them will come in salivating, urinating, defecating, just delirious, and sooner or later they're going to have seizures because you can't stop the muscles. Okay, that's called sludge syndrome. Sludge stands for, the S is want to get the door. The S is salivation. The L is lacrimation, crying. The U is urination. The D is diarrhea. The G is GI pain or pain, and the E is emesis or vomiting. Okay, and you'll know why those particular sets of symptoms happen when we get to the neuro section and talk about the sympathetic and parasympathetic nervous system. Okay, because most of your rest and digest systems run on acetylcholine. Okay, whereas your fight and flight system, the sympathetic system, runs on adrenaline and norepinephrine. So you get a lot of GI symptoms when you have acetylcholinesterase poisoning. Okay, and if it's bad enough, you go into seizures and you die from suffocation because your diaphragm won't work. Your diaphragm seizes up and you can't breathe, so you die. Okay? And it doesn't take much; just half a drop of some of this stuff will kill you. So that's what they released in the Japanese train system a few years back. That cult that put the sarin gas in the train. So. But you will see it most likely from accidental poisonings in the ER with little kids who are drinking a bottle of, insect spray or something, or something like that. They're really trying to get away from those and use other toxins because these are so deadly, and nasty. The antidote for that, because I've got too much acetylcholine in here, the antidote for that is atropine. Okay? Atropine is sort of a poison in itself because atropine is a pretty nice little round little molecule, and what it does is block the pore. Okay? So if I've got too much acetylcholine, I give atropine, it blocks the pores, and then the effect goes away. Okay? So that's why soldiers carry around atropine to stick themselves in the leg if they get nerve gas poisoning. Okay? And that's why you give it atropine in the hospital to people who had insecticide poisoning. Okay? So that pretty much sort of explains the whole process now. Okay? From the positive charge triggering influx of calcium, which makes the vesicles release acetylcholine. That opens up the chemically gated sodium channel, which opens up the voltage-gated sodium channel, and then the wave starts. Okay? And because the voltage will shut these things on and off, okay, it's just, it is a wave that goes, and behind it's going back to normal. Okay? And so, now there's a lot of things, just like little nerve gases. If you want to play around with things that can go wrong. We know all these things are proteins, right? All the membrane, integral pores, they're all proteins, which means they're all made by genes. They're all coded in your DNA. So, and not all of them are one protein either. They're complexes of proteins that form these pores and things. So now, you've got about, how many genetic disorders do you think you can have based on screwing up with any of these proteins? Okay? You can screw up with the thin filament proteins, the myosin, the walkers, okay? And you get muscular problems, okay? Weakness. If it doesn't kill you first, okay? They're usually fairly mild disorders, otherwise you wouldn't be born, okay? What if you screw up with the way this thing attaches to the cell? If it couldn't attach here, then the cell, the sarcomere might contract, but the cell doesn't. Okay? It has to be attached to the wall, right? So then the whole cell contracts. Well, that's muscular dystrophy when that happens. That's the problem. It's a big protein called dystrophin, and when you have a genetic disorder, your sarcomeres don't attach to the cell, and you get very weak. Okay? And we know that's one of the most common causes of muscular dystrophy now. Okay? Wasn't discovered but not too long ago. Okay? So the protein that makes the winding guard, okay, that just happens to be called tropomyosin. Okay? Troponin is the name of the one that reacts with the calcium. Okay? So all these things have names. Myosin, okay, is the muscle walker. So that's why they call it the actinomyosin system. Okay? You've got actin and myosin. You've got tropomyosin, and troponin is the little trigger that takes the calcium. Okay? This little bag here, I haven't given it a name. This is what used to be in most other cells is the endoplasmic reticulum. Okay? In the muscle cell, you call it the sarcoplasmic reticulum. So it's not a magical thing that muscle cells have. They just use the endoplasmic reticulum to store the calcium. Okay? And so any calcium genetic pump problems here, genetic pump problems here, you've got hundreds of diseases that are related to these kind of things. Let's see. Let's talk about how if you wanted to just give somebody just to paralyze them so their muscles wouldn't work, well, what could you do with this system? Maybe not even permanently, just temporarily you want to paralyze them. Like in surgery, which we do every day to people. This is how it works.
Atropine, isn't it?
Sorry?
Give them a shot of atropine.
Well, that'll dry them up, or the atropine doesn't really paralyze you too much. But like I say, it's a partial blocker of the sodium, chemically gated sodium channel. Okay, it's you get a lot of it. It can be toxic, and you can die from it, from seizures and things like that. But I'm certain not seizures. It's more respiratory problems and brain delirium, things like that. Atropine is what's in the little flowers. They call them the trumpet flowers. Okay, people make tea from it. Usually some teenagers are out there. That's what is in atropine. It's atropine that's in that stuff. But it's also brain or synapses that it affects. So you get some delusional and hallucinogenic kind of effects with it. Well.
So what do you give?
The acetyl.
Well, I'm not necessarily talking about what do you give. I'm talking about how.
Well, would you want to?
What do we want to do to do that?
Would you want to block the acetylcholine?
I didn't say increase the sodium.
Increase the sodium?
Or decrease it one.
Well, yeah, you're kind of thinking of, terrible consequences of major sodium or potassium imbalances. Yeah, that's why it's important for IV therapy to keep people's sodium levels right, potassium levels right. That's why docs are always freaking out about the electrolytes and things like that, because you have to keep this system working, okay? If you don't have enough sodium in your body, you can't keep this gradient, okay? And it's usually in a situation where you're dying of dehydration and you're salt depleted and things like that. But yeah, let's assume that we're taking a normal person and with normal levels and things like that. What can we do to this system to make them just go paralyzed?
Shock the nerve.
Shut
down any one of the channels?
Yeah,
Almost shut down any one of them without making it fire,
right.
okay? We could shut down this channel, okay? We could shut down this one, okay? And you're not going to get a depolarization if we shut down the voltage-gated channel or the chemically gated channel. And there are drugs that do that, okay? Like I say, a lot of the drugs that you get before surgery to paralyze you shut down the chemically gated sodium channels, okay? And then they gradually wear off, okay? And then you come out of that paralysis, okay? But if you gave them something that was permanent, that actually covalently bonded to the protein and shut it down, then you kill them, you paralyze them for good, okay? A lot of your painkillers, like lidocaine, all the stuff they give you to numb your teeth up there, all they do is block the sodium channels. And so it can't fire. But it's not only the muscles that you're paralyzing in that case, which is why you get the droopy lip, because your orbicularis oris is paralyzed. Okay? That's the muscle. But you also notice that you're not feeling any pain. Okay? That's because you're doing the same thing to the nerve. You're paralyzing its sodium membrane or channel. Okay, so that's why these are so similar, and this is why I put them together in this lecture, because this is really supposed to be the muscle section. Okay, this is supposed to be part of the neuro section, but they work so well together that now you've done the first chapter of the neuro section, and you don't need to do anything. Okay, so we'll get through it, but just so many things. Anti-seizure drugs work by changing membrane potentials, changing the way the sodium, or chloride sometimes. You know, if you let chloride into a cell, there are chloride gated channels. Okay, if chloride can get in, what's going to happen?
It'll offset the positive charge.
Well, this is not here. The normal voltage is negative, right, on the inside. So if I add more negative voltage, I'm not starting at minus 70 millivolts, I'm down here, okay? And I have to get way up here to get to my threshold before it'll fire, okay? So what most anti-seizure drugs do is fool around with chloride channels and things like that, and lower the or lower the resting potential so it's harder for the nerve to fire, okay? Most of your drugs, like your sleeping pills, you know, phenobarbital, Valium, Xanax, all the similar anti-anxiety drugs like that, they tend to work on these channels, blocking the entrance of the sodium or adding chloride to lower the threshold, okay? Because if anxiety is too much of your brain firing, what you're doing is kind of just taking a total brain depressant, okay, and slowing things down. That's why it works. Your antidepressants work, but not on acetylcholine. They work on the ones that are in your mood centers, okay? Things like serotonin and dopamine. Those are transmitters in the brain, just like acetylcholine is here. But with the muscle, acetylcholine is the only one you have to worry about, okay? But in the brain, there's dozens of them, okay? And all these little channels, you block them with antidepressants, and sometimes one antidepressant will block two types of them, and one it won't. That's why not everybody responds to the same antidepressants. Sometimes you got to try 2 or 3 before you get one that works for you.
Is that why sometimes when you take, like, because I know I'm bipolar and I take medicine for it, and the medicine that I was on, like, it made me, like, real tired and, like, depressed almost, like I couldn't, like I was sad all the time.
Yeah. You can see exactly what's happening. You're blocking the wrong channels, okay? What Prozac actually is it blocks the reuptake of serotonin. The nerve releases serotonin into the synapse, and then it has to reuptake it to use it again after it's sent the signal. Okay, the things like Prozac and Zoloft, those antidepressants block that reuptake, so the serotonin stays in the synapse and keeps stimulating the nerve, which is what the problem is. You're not stimulating it enough. Okay, so that it prolongs the stimulation. But there's other drugs like Wellbutrin that get norepinephrine channels in the brain and other antidepressants, so they all work a little differently. And not everybody has the same depression, because not everybody has the same problem with their transmitters and their channels. Okay, so it's not a one-size-fit-all problem. So,
I have a question, Dr. Ursula. What is that process? If I want to look it up on YouTube, what would that process be?
well, it's kind of two separate processes. You can look at muscle cell contraction.
Okay.
You can look at action potential. Okay, and I'm going to show you one on YouTube that's just labeled action potential.
Oh, okay.
We've got 20 minutes, so we've got plenty of time. You can look at neuromuscular junction function. Okay.
That's kind of fun to say.
We'll talk a little bit more about how the nerves actually kind of affect each other when we start talking about the brain and that sort of thing. But when you have a nerve, I'm just going to make it like this, it has an axon. Okay, it sends out an action potential down the axon, which is going to affect another nerve. Okay, which is going to send out an axon and affect another nerve. Okay. Okay, and it may not. It may have little bodies called dendrites, and then there's another nerve over here that's going to affect the dendrite. Okay, these are things that are bringing in that positive charge into the main body. Okay, and when the positive charge builds up right here as what's called the hillock, then this thing will fire and charge. It only fires it down an axon. It won't fire it down dendrites, and there may be thousands of dendrites. It's estimated that the neurons in your brain, the pyramidal neurons in your brain, each one of them have 5,000 to 10,000 connections with other nerves. And there's 100 billion of them. So that's the complexity that we're talking about. You see pictures and it's just mind-boggling. But what I wanted to show here was some nerves are sending signals that are making that more negative. So they're inhibitory neurons. This could be an excitatory neuron. It's trying to excite this thing to go, and maybe if this is at rest, that would be enough to get this neuron to fire a signal to the next nerve. But you've got other nerves that are inhibitory that are increasing that negative charge, okay? And those are things like GABA neurons that are inhibitory. And so your brain is like a computer. It's adding up the charge and deciding when it's going to fire based on the input it's getting. So some are negative, some are positive. Okay? So every neuron is like a computer, and it's got 5,000 connections. Okay? So it's unbelievable. Okay? All right. I'd love to talk about this some more. If anybody has any questions, please. No? Just overwhelmed? We'll do it again. Like I said, you're not going to get it the first time. I didn't. Matter of fact, I don't think I got it in the first class. I got it in another class I took later. The first time, I don't understand it.
This is one cell. That's another cell.
This is all one cell.
This is all one cell.
But in order for,
This just happens to be the T-tubule that's in the muscle cell.
That's in the cell.
Yeah.
So this is all one cell.
Yeah,
Okay.
this is all one cell.
but in
Gotcha.
order for muscle contraction to take place, it has to start at the nerve.
It has to start the nerve.
Nerve.
And that's the interesting thing. Every single one of your muscle cells has a nerve connection. Okay? And
Every single
one. Every single one. Sometimes if your nerves, let's say the muscles in your eye,
Okay.
they have to be controlled really tightly, really
Right.
perfectly. Okay, I can control my eyes the way they move, just very finely. So it might be one nerve cell that controls maybe 5 nerve muscle cells. Okay, it's
Okay.
got 5 axons.
Okay.
So that one nerve is controlling just
So it's
5
not
cells.
there's a nerve for every
No,
muscle cell,
no.
but every muscle cell is connected to a nerve.
To a nerve,
Okay.
right? But the ones that are finely controlled are connected to very few neurons. like your eyes.
Okay.
They think it's about 5 to 10 fibers for one nerve, okay? In the vastus lateralis of your thigh, probably hundreds of muscle fibers are controlled by one nerve.
One nerve, okay.
Because I don't need to control my thigh that finely.
Right.
Okay? So one nerve, they give it, the joint duty. Okay?
Okay.
But the nerves in your eyes, those muscles are finely controlled, and much more finely controlled in your hands. Okay, there's a much bigger slice of your motor strip in your brain for your hand than there is for your leg or your foot. As a matter of fact, it's probably about equal. The hand in the brain, the section that controls your hand, is about the same size as the section that controls the whole bottom of your body. Okay, so the hands are really finely controlled, as well as the face, the lips, the tongue.
Yeah,
Very fine control.
What happens if you had an injury and you had nerve damage? Let's say you had nerve damage to your calf muscle.
Yeah.
There would be those nerves that were affected that were injured and have died off or something like
Right.
that. But
Right.
the rest of the nerve cells in your muscle cells are fine.
Oh yeah, it'd be fine.
So the damage would be here.
Local damage, anything below the injury.
Mm-hmm.
Yeah, yeah. And that's why if you break your back, you get everything below the injury.
Mm-hmm.
You know, if you cut off the cord, all the nerves that are coming out of those intervertebral foramen and going down in the lower part of your body, they're now cut off, dead. You turn the switch, they're off,
Mm-hmm.
and you don't move, and you don't feel anything.
They don't grow back?
No, not usually.
No.
That's what they're working with stem cells to do, to try to grow those things back. But most of the time you cut a nerve, it's gone.
On the gates, you have just one chemical gate, one voltage gate
No, there's thousands of them.
of
Okay.
all of them, or
Yeah, all of them.
I
Yeah,
mean-
yeah. This is it's like looking at a field of them. Okay, and that nerve just dumps the acetylcholine on, and hundreds of them will fire. Okay, and then that positive charge then will affect the voltage channels, and there's hundreds of those around. Okay, so you get this big wave of charge moving out across the muscle, but immediately after the wave, everything goes back to normal.
So it's not just one chemical gate and then a bunch of voltage gates. It's a bunch of chemical gates and then a bunch of voltage gates.
Yeah, but the chemical gates are only on that motor end plate. So
Okay.
it's like a special spot on the muscle
Okay.
where the nerve is attached.
Okay.
All the rest are going to be voltage.
All the rest will be
Okay,
voltage.
that's
Okay.
what I was
Okay,
trying to figure out.
okay. Yeah.
So when you have a stroke, what is it the voltage that's more affected or the chemical gates are more affected?
Well, you basically kill them all.
Kill them all?
Yeah, the nerve cell dies from lack of oxygen usually. Okay. So they're just not going to fire anymore. They just deteriorate like a scar. And the cells inside the brain just sort of eat it up and become sort of this scar. Okay. But they're not firing anymore.
What if a person has like TIAs, like mini strokes all the time?
That's just the transient oxygen problem. You know, so it's kind of like holding your breath just long enough to kill a few brain cells and then coming through, you know. So you can have.
But it does kill like the nerves.
Oh yeah. Well, it's like I say, it's bound to kill a few, but most of them are on the verge of dying. But the oxygen gets back, and then they recover. Okay, so then you might get full function back after TIA. Transient ischemic attack is what you call partial strokes, or strokes that last less than a few hours to a day or so. But a real stroke, it's the oxygen that doesn't get to the nerves, and the reason is the mitochondria are shut off with no oxygen. This is the whole how you die. You know, what is life? You know, you got to shut the oxygen off. And the mitochondria is what dies first because they can't produce ATP now. And now everything else that relies on ATP can't work.
Shuts down.
And then everything shuts down. Okay.
Quick question. So when we're sleeping,
Yeah.
is this all taking place or it's kind of like at rest itself?
No. You'd be surprised how active your brain is. You know, just the act of dreaming is
Enough
more
to keep
complicated
them
for the
not
It's
brain.
like when you're asleep and you
Then-
think you're kicking something and you wake up and you're
Yeah.
shaking
And
your
you are,
legs.
yeah. It's all working. Yeah. The thing that shuts down is your perception of consciousness. Okay, but your brain is still working just like always. Okay, I mean there are certain parts of the brain that are resting more, but it doesn't shut down.
Right, right.
So when you're dreaming and your brain's actually still working,
Oh yeah.
and you're dreaming that you're making chemical solutions and breaking them down. Can you actually figure them out while you're sleeping?
Sometimes, yeah.
Because I would wake myself up. I was studying so much when we were on that part.
Yeah.
I'd wake myself up and say, "Stupid, you're not going to figure it out. You don't know it. Why are you dreaming?" Then I go back to sleep and think it was the dumbest thing.
There are plenty of stories where people have come up with creative ideas in their dreams,
Wow.
even in the sciences.
Wow.
The guy who discovered that benzene was in a ring. Nobody ever knew about rings before. Saw it in a dream.
Yeah.
Wow.
Oftentimes, 5 o'clock in the morning, I'll have a great design idea, and if I get up fast enough and run to the computer before it's left my
Yeah,
brain,
Yeah.
then it's good. Even just a chicken scratch it. Yep.
Well, the big question now is, and with brain science, is what is consciousness? You know, we know how all the muscles, how all the neurons work. We know the transmitters. We just don't know how the structural stuff is capable of giving us a perception of being me. You know, what is consciousness? You know, it's like you can understand all the nuts and bolts, but what all of a sudden makes something self-aware and conscious? There's a lot of research going on in how memories are made. Okay, apparently the more the neuron fires, the more calcium channels you get on the neuron. And it becomes more likely to fire again. So that's sort of what memory is. You know, when you cross over and that neuron gets stimulated again, it fires more likely, and that's how we keep memories. Okay, but it's more complicated than that, obviously. But it's all about making new synapses in the brain to form memories and to think things through. Okay? And the ones that are more strengthened and have more neurons in that pathway are stronger memories. Okay? And obviously our brains are rewiring this stuff all the time because we're not like tape recorders. We don't have a permanent memory. That memory will change over time. And you can change it just by looking at an old picture. Somebody tells you, remember we went here, and all of a sudden, you know, your memory is, you think you have a memory, but you really don't. You know, our memories really are screwy. They can be changed just by suggestion. Remember when I was wearing that red jacket? Oh yeah, I never had a red jacket.
I had my husband convinced for a whole year that he was a year older than what he was. I really did. It was hysterical. He wasn't very happy when his birthday came up and he realized that I'd been lying to him.
Well, that's, I mean, I find it fascinating, gang, but this is kind of the basics of, you know, so much of medicine. And what you're trying to do with fluids and electrolytes, and then you're trying to keep these systems working, keeping these levels in the right way, in the right place. Let me see if I can show that little video. I'll let you go. Like I said, I know this is.
That's true.
I know this is a brain full, but we'll be going over it. I've got a lot of this on the slides, so we'll be talking about it all again. But if there's one thing I want you to remember 5 years from now, it's this stuff. You can forget the tibia and fibula.
I've already forgotten.
27 and he's like, and I said 11 because every time he was about to say 27,
about bones, I was
and I just told him he was a year older. He believed it. It was hysterical. But you don't think about those things. Well, he doesn't.
Right. No, they used to go.
Well, oh, that sucks. Ooh. Ow, that sucks. That hurt me.
Did it right on.
Ooh.
Here we go. Did somebody hit the lights?
Is it that one or is it the wall?
I think it's over to the door. I think
Yeah.
it's that one.
Thank
The
you.
process of
Watch your step.
excitation, contraction, coupling consists of all those processes involved between the time of generation of the action
This is very
potential on the
simple.
muscle
It goes
sarcolemma
over everything.
and the actual contraction. As the action potential reaches the neuromuscular junction, vesicles containing acetylcholine fuse with the membrane of the nerve terminal, and acetylcholine is released into the cleft. Acetylcholine molecules rapidly diffuse across the cleft and bind to the acetylcholine receptors. Binding of acetylcholine to receptors immediately opens ion channels. allowing sodium ions to rush into the muscle fiber and potassium ions to diffuse out. Acetylcholine is then split to acetate and choline by cholinesterase, resulting in inactivation and closure of the channel. The initial flux of ions due to opening of the channels brings the membrane potential to threshold, causing the voltage-gated sodium channels in the vicinity to open. The resulting action potential is propagated over the entire surface of the muscle and also courses into the T-tubule system. As the action potential encounters DHP receptors in the T-tubules, it causes opening of the ryanodine calcium channels of the lateral sacs, thus allowing calcium ions to diffuse out of the lateral sacs and into the region of the myofibrils. Calcium then binds to troponin, resulting in movement of the tropomyosin-troponin complex away from myosin binding sites on actin. The contraction process continues as long as action potentials continue to reach the neuromuscular junction, thereby keeping the concentration of calcium elevated around the myofibrils. To end the contraction, calcium must be actively transferred by the calcium pump, calcium ATPase, back into the lumen of the lateral sacs. This pump utilizes ATP as a source of energy. This process reduces the concentration of calcium in the vicinity of the myofibrils, such that calcium dissociates from troponin. Tropomyosin again covers myosin-binding sites on actin, and the muscle relaxes.
There you go. There's a lot of that sort of stuff on YouTube if you want to.
And what was this called?
Let me see this. That's called action potential.
Action potential.
Look for this picture when you do
Consists
it.
of all those processes involved between the
One minute. Oh, I just thought of one little extra tidbit. No, it's not. There's PBS. Botulism. Everybody knows that that's a toxin, poison.
Yeah.
They make Botox with it. You know, actually, poison is in the dose. You know, some things are good in little doses, and poison in big doses. Botulism toxin screws up with this protein right here that lets the vesicles release, so they can't release. That's why the Botox relaxes the muscles, because they can't fire. They don't have any acetylcholine. So, all righty.